Hello, everyone. I'm a former PhD candidate at Lincoln University under supervision of Nik, David, and Keith. And so if you have any question about my PhD work, this talk, or anything related to it, please don't hesitate to reach out to me directly or to any one of the three if you have any questions or want to know any detail. So yeah, I'm very happy to be here. And when David asked me to virtually present one of my PhD studies, because I was actually prepared that I couldn't attend because I have graduated and left New Zealand a few months back. And so I thought I probably cannot come. But yeah, David made it possible that I can present here some of my work. So the study I will present investigated the effects of plant species on carbon rhizodeposition in nitrogen-rich grasslands. So this is a conceptual diagram with the coupled cycles of carbon and nitrogen, which I will explain very briefly. So the carbon cycle is indicated here with blue arrows and starts off with atmospheric carbon dioxide, which is assimilated by the plant through photosynthesis. The plant respires some carbon, but that's of minor importance here. The majority of this carbon that is assimilated is going into biomass generation of the plant and is released through the roots into the soil. And this is what we call rhizodeposition. And this is a term that you should remember because I will use this probably, 500 times throughout this talk. So this rhizodeposited carbon is usually highly available for uptake by microorganisms dwelling in the soil, and so it can become part of soil organic matter, but it can also be respired through microbial activity back into the atmosphere. Microbially driven processes here are written in red fonts, by the way. The processes that regulate soil carbon cycling are typically closely coupled with the nitrogen cycle, which is indicated here in green arrows. So soil organic matter usually contains a really high amount of nitrogen, and this can be released to mineral nitrogen through microbial activity, and this is what we call nitrogen mineralization. This nitrogen can then either be taken up by the plant or undergo nitrification, and thereby increasing the risk of nitrogen losses through emissions and leaching. But mineral nitrogen can also be incorporated into soil organic matter through microbial nitrogen immobilization. And so whether nitrogen is preferentially immobilized or mineralized depends, among other factors, on the concentration of carbon relative to nitrogen, or what we call the carbon to nitrogen ratio. So this is just a very simplified conceptual diagram, and I will come back to this. But so I try to use this concept to apply to a livestock grazing system where we have high inputs of nitrogen from urine patches. And so based on this concept, the nitrogen addition would lower the carbon to nitrogen ratio in the soil and so stimulate microbial nitrogen mineralization. But an increased availability of nitrogen would also remove nitrogen limitation for the plant, and so plant uptake should increase. When the plant grows better because of better nitrogen, it probably photosynthesizes also more, and with that also releases more carbon through rhizodeposition into the soil. And this would then increase the carbon to nitrogen ratio, which would favor nitrogen immobilization. So there are a lot of arrows here, and actually it's quite a controversial theories that are out there and are being discussed. So it's not really clear what is actually driving the processes that regulate carbon and nitrogen cycling in the system. And yeah, during my PhD, I tried to look into these processes. So these are some of my previous findings where I investigated the effects of different grassland plant species on nitrification rates. So as part of that study, I measured the concentration of water extractable carbon in the soil of these plants. So the plants you can see here on this bar plot on the x-axis and the water extractable carbon on the y-axis. And I had treated these plants with two different rates of nitrogen. One was more like a low and the other one like a high, to see how the plant and the soil system responds to this. So first, one of the results that I saw was that the concentration of this water extractable carbon fraction was increased when a plant was present compared to an unplanted control, which is indicated here. And this was mostly evident when the growth conditions of the plant were improved in the high nitrogen treatments, which are here the filled bars, or the darker gray bars. Another result that I want to point out here is that although not statistically significant, but there were slight variations in this water extractable carbon between the plant species. For example, the plantain here stood out a little bit because of the overall higher concentrations compared to the other plants. So although it's not statistically significant, but it is something to keep in mind. Finally, I was able to observe a significant negative relationship between water extractable carbon and nitrification activity, which means that the potential for nitrogen to be nitrified was reduced the more of this carbon fraction was there. And so that's a quite important result based again on this conceptual model that I just showed. I will come back to that in a second. But one big shortcoming that this study had was that I couldn't measure where this carbon is coming from. Is this released through the roots? Is this coming from soil organic matter or any other processes? I couldn't measure, and so this is where I wanted to look into more detail and actually, try to measure rhizodeposition. So, briefly coming back to this conceptual diagram, there is a whole lot of stuff going on, and it is just too much to study all of these processes in just one study. So I broke it down into just one aspect that I wanted to investigate, and that is whether carbon rhizodeposition increased proportionally with increasing nitrogen addition. And of course, whether this effect is different between different plant species. And I also wanted to know what is actually happening in the soil with the rhizodeposited carbon once it is released. So to measure this, to investigate this, I set up a microcosm experiment under controlled conditions with two plant species. I used the perennial ryegrass as the typical pasture species in New Zealand and compared it to plantain, which may have a higher capability to release carbon into the soil based on my previous observations. I treated these two plant species with an increasing rate of nitrogen, which are somewhat typical for urine patch concentrations of grazing livestock. On these pictures you can see, or I have taken these pictures actually at the very end of the experiment when the plants had already grown under these nitrogen treatments for several weeks, but you can see that in the lower nitrogen treatments, the plants already experienced some level of nitrogen limitation, whereas at the higher nitrogen levels, the plants still look really green and lush, and they are doing pretty well. I will also, throughout this talk, I will use this red and blue color here for the different plant species, so you will definitely see those again. During the experiment, I monitored net ecosystem carbon dioxide exchange, as you can see on these pictures here. At the end, I pulse labeled the plants with carbon-13 enriched carbon dioxide for several hours and then destructed the microcosms and measured the carbon-13 concentration in several plant, soil, and microbial pools. For example, I did some PLFA stable isotope probing here, which to my knowledge, nobody in New Zealand has ever done that on an environmental sample. Let's have a look at some of the results. So this is the composition of the soil microbial groups after non-metric multidimensional scaling. And it's separated on the right for the different nitrogen addition rates and on the left for the different plant species. Both treatments had a significant effect, but I want to point out the very clear effect of the plant species here on the left. So you see that there's a very clear separation of the microbial group composition between the plant species. And this really highlights that each plant species fosters a distinct microbiome in its root zone, which is quite likely related to rhizodeposits. And yeah, this is what we will look at next. So here on this plot, you can see for each rate of nitrogen addition on the x-axis, the concentration of rhizodeposited carbon-13 within the water extractable carbon fraction on the y-axis. I should also point out that the y-axis is on a log scale. So the fact that it is the carbon-13 enrichment means that this carbon can only be from recently assimilated carbon, and with assimilated I mean photosynthetically assimilated carbon, and can only come from the plant roots because it was already in the soil. So this has to be rhizodeposited carbon. It cannot come from any other source. You can see here on this pattern very clearly that the concentration of this rhizodeposited carbon increased for both plant species, mostly proportional with increasing nitrogen addition. You can also see that the plantain released overall more carbon into the soil than the perennial ryegrass for the labeling period. So if you remember back to the conceptual diagram, if we follow the assumption that rhizodeposited carbon is usually highly available to microorganisms, we would see, or we would expect to see, a similar pattern for microbial uptake of rhizodeposited carbon. So these are the PLFA stable isotope probing results. And for each panel here, which represents one microbial group, there is for each nitrogen addition rate on the x-axis the concentration of rhizodeposited carbon-13 that was incorporated into the microbial biomass of the certain group on the y-axis. The y-axis is in most cases on a log scale. So the results show that microbial uptake of rhizodeposited carbon depended on one hand on the microbial group, shown here by the different patterns of uptake dynamics, and but on the other hand, there was also an interactive effect between the nitrogen addition rate and plant species. To make that a bit clearer, I will zoom in on one example. I picked the gram-negative bacteria because this microbial group usually favors very labile carbon sources, for example, rhizodeposits. So based on that, we would assume, or we would expect, that gram-negative bacteria would benefit from increased carbon rhizodeposition and increase its consumption when there is more of it available. But we actually see that only for the perennial ryegrass here, where there is a strong increase in rhizodeposited carbon uptake by the microbial group, but for plantain there's actually a decrease in microbial uptake, which is a bit controversial given that on the previous slide we have seen that the amount of carbon rhizodeposition was overall significantly higher for the plantain than for the ryegrass. So this is obviously not reflected in the uptake of rhizodeposited carbon by this microbial group. And this is a similar pattern for all of the microbial groups, where the response in microbial uptake of rhizodeposited carbon to this increasing rate of nitrogen addition is much less pronounced for plantain than for the ryegrass. What I mean with that is that the slope of these lines that I'm showing there is often much steeper for the red line, so for the ryegrass, than for the plantain, where it is much less affected. So this synergistic effect between the plant species and the nitrogen addition rate can have significant consequences for carbon cycling. For example, to put that into an applied context, where high nitrogen addition from a livestock urine patch may cause an increase in rhizodeposition for a certain plant species, this doesn't always translate into increased microbial uptake of rhizodeposited carbon. Also, which microbial group benefits from this increased rhizodeposited carbon plays an important role because different microbial groups just influence the carbon cycling in different ways. For example, in this study, we have seen that rhizodeposited carbon from perennial ryegrass was increasingly allocated to microbial groups that are associated with fast carbon cycling, and this suggests there's a reduced potential for long-term carbon storage. But for the plantain, it was the opposite. Rhizodeposited carbon was preferentially allocated to microbial groups that are usually associated with slow carbon cycling, which may suggest a higher potential for long-term carbon storage. So it's quite obvious that these processes are very complex and that in these high nitrogen systems that we are working with, we always have to consider also the dominating plant species that is grown if we want to make any predictions about carbon cycling in these systems. And with that, I would like to end my talk and to acknowledge everyone who has been involved and helped with this study. I also added my contact details in case you would like to reach out or have a question. Please get in touch at any time. Thank you.